André Huss Eriksson, Pharma. Treating skin diseases. On target, in time.
Hello. Today I'd like to take you into the engine room of the drug discovery process for a topical drug. Next, please. And what you can see here is a lot of stuff going on. Very early on in the narrow arrow, you have all the early in vitro biology going on, early chemistry, and where you, as my colleague Paola Lovato explained earlier today, we get a very detailed in vitro pharmacological mechanism of action knowledge about our new drugs. But eventually we'd like to put it into the clinic, and in order to get some really early data on how these drugs will penetrate the skin and work, we use these ex vivo skin pharmacokinetic models that I'll show you. What is it? We use fresh human skin. It has that we keep alive in culture, and that allows us to rank new drugs and also new formulation principles with regards to how much gets into the skin and how much is the target engaged. So this is what I'm showing you here as a dose response and a concentration, skin concentration response relationship of 4 different compounds. Some of them are reference drugs, some of them are our new compounds, and what it allows us for is then to get confidence that our new candidate will have a similar target engagement as the reference drugs. And what we also are able to do is look at different formulation principles. So what you're seeing here is a dose response. Now the response here is down is good, before up was good, now down is good. Where you have 4 different strengths of an ointment that gives a nice dose response. You see that the 30 milligram per gram ointment gives the largest response. But the ointment is a very poor formulation in terms of patient compliance. So if we switch to a solution, which is much nicer formulation, we can potentially reduce the dose by a factor of 10 and still retain the same target engagement. So this is also an example where instead of using target engagement as we do on the upper panel for calcipotriol, then for betamethasone dipropionate we can look at the metabolite, because if it's metabolized in the skin, it's probably also available for action. So in short, we use human skin explants as relevant and valuable tools in topical drug development. We can rank new compounds for drug candidate selection, and we can aid selection of new formulations for clinical development. So thank you for your attention. Shoot me an email. I'll be running off shortly after this is over, so thank you.